Very good morning to all of you. Yesterday's part, what I have posted was associated with the MCQs for microbial metabolism. Now, in the previous lecture, we have gone with degradation, the basic part of degradation of amino acids. In this degradation of amino acids, there is one part that is called as urea cycle, which plays an important role in the degradation of amino acids. So let us understand the urea cycle. In eukaryotic organisms, the ammonia deposited in the mitochondria of hepatocyte is converted to urea in urea cycle. This pathway was discovered in 1932 by Hans Krebs and Kurt Henseleit. So Hans Krebs is the same person who has invented the citric acid cycle. We have already gone to this citric acid cycle in some previous lectures. Urea production occurs almost extensively in the liver. Now, why this urea that has been generated and why urea cycle has to run, because urea in a higher level is said to be toxic in the nature, and therefore it has to be degraded through the urea cycle. Next, now this is the urea cycle. In this urea cycle, if you see, there are a number of amino acids you can see which come play or which go in the urea cycle and get degraded into various different products. And there are certain key enzymes of this particular pathway which helps the mechanism of the urea cycle to run and degrade the urea. And ultimately, the urea has been liberated in this particular format. What is the step one? In the first, amino acids to enter the urea cycle is derived from ammonia in the mitochondrial matrix, NH4. Then H4 generated in the liver of mitochondria is immediately used together to CO2 as HCO3 produced by mitochondrial respiration to form carbamyl phosphate in the matrix. The ATP-dependent reaction is catalyzed by carbamyl phosphate synthetase 1. See this. From ATP to bicarbonate, ATP is released. Carbonic phosphoric acid anhydride is being generated. That is get converted into carbamide. This carbamide utilizes another ATP molecule to form carbamyl phosphate. So this is the first step in the urea cycle. Step second, the carbamyl phosphate functions as an activated carbamyl group donor enters the urea cycle. Carbamyl phosphate donates its carbamyl group to ornithine to form citrulline, which with the release of EI, the reaction is catalyzed ornithine transcarbamylase. This is ornithine that has been generated in the previous reactions, and it is now get converted into citrulline. Citrulline passes from the mitochondria to the cytosol. So citrulline is a particular kind of product which from the mitochondrial matrix gets converted or gets passed from the mitochondrial matrix to the cytosol. Second amino group now enters the aspartate generated in the mitochondrial by transamination and transported into the cytosol by condensation reaction between the amino group of aspartate and urido, that is carbonyl group of the citrulline to form arginosuccinate, catalyzed by arginosuccinate synthetase. Here, citrulline now gets converted to arginosuccinate. So two amino acids have been joined to each other to form arginosuccinate. Arginosuccinate is then cleaved by arginosuccinase, the enzyme, to form the free arginine and fumarate. Fumarate enters mitochondria to join the pool of citric acid cycle intermediates. So arginosuccinates now have been degraded, succinate get converted to fumarate, which is a very basic reaction. We have already seen this part. And arginine has been relieved. This fumarate which has been generated now gets attached with the citric acid cycle, and ultimately it gets converted into the citric acid cycle. So arginine, which is left behind, cleaves arginine to yield urea and ornithine. The ornithine is transferred into mitochondria to initiate another round of urea cycle. So this arginine gets converted to the ornithine once again, and urea has been liberated in this particular format. Again, ornithine, which undergoes the same cycle of the urea cycle, wherein the urea will be liberated once again. So this is the link between the urea cycle and the aspartate shuttle of the citric acid cycle. See this urea cycle, arginine to ornithine, urea has been released here, and this ornithine gets converted to citrulline, Cytol to arginosuccinate gets converted to fumarate, fumarate to malate, and malate again been funneled into the citric acid cycle, TCA cycle. Even the fumarate which is generated in an excess amount, instead of converting, it gets directly to fumarate. From here, the citric acid again been funneled. Citric acid, the aspartate which has been produced, this aspartate goes and joined to arginosuccinate. Arginosuccinate gets converted to ornithine, and both these cycles run as accordingly with with coordination with each other. Correct? So that's all for today's work. Have a good day.